So far, we've covered how to design and extract nDNA and how those signals let us read model-level operations, merging, distillation, quantization, pruning, through curvature, path length, and belief steering. Now let's shift gears and introduce neural genomics. Neural genomics is a new paradigm for studying the evolution of artificial cognition. It asks what gets inherited, what recombines, what collapses under compression, and what new traits emerge when two lineages meet. In place of weights went up or down, we talk about ancestry, variation, and fitness of ideas inside a model. Why this matters? Accuracy only tells you whether a model was right or wrong. Neural genomics explains how it learned to think that way, and whether that thinking will hold under pressure. With nDNA as our instrument, we can trace family trees of models, preserve epistemic richness during compression, and design fusions that create capability without chaos. In short, nDNA is the lens, neural genomics is the map. Let's use both to chart where our systems come from, where they drift, and how we guide the next generation, not just to be right, but to reason well. Within neural genomics, we unveil a diagnostic kit, 7 readouts that reveal how a model's mind evolves. NHD, neural Hamming distance, flags low-level architectural drift across layers, great for spotting silent mutations and training scars. NGDI, genetic dissimilarity index, measures layer-wise geometric divergence between models, a clean read of lineage distance and epistemic separation. Enkaryotyping, semantic chromosomes clusters latent semantics into chromosomal blocks, revealing which concepts travel together and how modules inherit. NTEDS, trait dominance scores, tracks prevalence and dominance of semantic traits, surfaces ideological narrowing or cultural imprinting. NDIV, directional inheritance vector, shows where offspring features actually flow from each parent, a vector field of epistemic lineage. NCCL, cultural conflict level, quantifies tension in merged spaces, the geometric signature of cross-culture dissonance and friction. NEPI, neural plasticity index, reads adaptive capacity under fine-tuning or perturbation, the pulse of learning flexibility. Together, these diagnostics turn nDNA from a lens into a dashboard, so we can monitor divergence, inheritance, conflict, and plasticity as artificial cognition evolves. In this video, we will show you glimpses of each of this metric in action. Neural Hamming distance, NHD, is a layer-wise measure of how often the sign of a model's activations flips. Think of it as a bit flip rate inside the network. Low NHD means the internal code is stable. High NHD means the code is jittery or being rewritten. Purpose of NHD: to pinpoint where cultural fine-tuning, merging, or other edits introduce instability, so we can regularize, freeze, or reblend those layers before the model becomes brittle. Biological inspiration: Hamming distance plays the role of a mutation scanner in genomics. Just as base pair changes reveal hotspots of genetic drift, activation bit flips reveal hotspots of neural drift, places where the model's internal genome is mutating under pressure. How to read the 3D tube? Depth on the x-axis, layer index. Curvature on the y-axis, spectral curvature, a proxy for how tightly the latent manifold bends. Thermodynamic length on the z-axis, total work the representation performs through the layer. Tube thickness, the NHD magnitude across prompts. A thicker tube means more bit flips. Colors, blue and red tubes are the two parents. The dashed purple trajectory is the offspring threading between them. What stands out in this example? Europe plus Asia. Early layers, tubes are slim, which means token and shape codes are conserved. Middle layers, roughly 24 to 27. Both parents thicken and the offspring passes through the fattest region, which marks a recombination zone where cultural priors clash and the code is most unstable. Late layers, tubes narrow again and the offspring hugs a stable corridor, which shows the child settling into a new, consistent codebook. Why it matters? Bulges in NHD mark actionable layers. Regularize there, soften the blend schedule, or freeze weights to prevent off-distribution wobble. If thick tubes persist near the top, expect brittle reasoning even when benchmark accuracy looks fine. Takeaway: NHD is your early warning sensor. Slim tubes signal steady lineage. Mid-stack bulges reveal where beliefs collide. Narrowing at the top signals a stable, interpretable offspring. For more, please refer to the book and the website for full definitions, settings, and additional case studies. Genetic Dissimilarity Index, NGDI, is a layer-wise measure of how far apart two models' internal geometries are. Think of it as a distance between their shapes of thought. Low NGDI means the manifolds are compatible. High NGDI means the two minds are taking different internal routes to the answer. Purpose of NGDI: to pinpoint where transfer or fusion will break down, so we can add adapters, retune normalization, or do targeted fine-tuning exactly at those layers before we merge, distill, or ensemble. Biological inspiration: in population genetics, genetic distance summarizes the separation between lineages. NGDI plays the same role for models. It is a lineage distance in representation space. How to read the 3D line with thickness? Depth on the XY floor. Layer index increases along the plotted depth axis. Curvature on the horizontal axis. Spectral curvature, a proxy for how tightly the latent path bends. Thermodynamic length on the vertical axis. Total epistemic work done through the layer. Line thickness, the NGDI magnitude across prompts. Thicker segments mean greater geometric dissimilarity. Colors: black dashed is the llama base, red is the Europe model. What stands out in this example? Europe versus llama base. Early layers: the red path is thin, token and shape encoders remain broadly compatible. Middle layers, roughly 23 to 27. Thickness spikes while curvature and length shift. This is the primary divergence zone where Europe's cultural priors reshape the manifold. Late layers, the path thins somewhat but keeps pockets of thickness around sharp turns, signaling residual differences in decision-level steering. Why it matters? Thick NGDI segments are the action points. Bridge them with small adapters, depth-aware mixing schedules, or local fine-tuning, otherwise merges will argue internally, distilled students will misinherit geometry, and ensembles will disagree of distribution. Takeaway: NGDI is your lineage distance meter. Thin means interoperable. Thick marks the layers where two minds no longer share the same shape of thought. For more, please refer to the book and the website for full definitions, settings, and additional case studies. Trait Entropic Drift Score, NTEDS, asks a simple question at each layer: which parent's latent traits are doing the talking? It computes an entropy-weighted dominance score over the trait bases learned during fusion. High positive NTEDS means parent AS traits are concentrated and in control. High negative means parent B dominates. Values near zero mean a balanced blend. NTDS is the depth-integrated version, one number that summarizes who dominates overall and by how much. Purpose: to expose asymmetric inheritance during merging or fine-tuning, where one lineage quietly takes over the internal discourse, so we can rebalance with adapters, data, or depth-aware mixing. Biological inspiration: in population genetics, drift and selection change allele diversity. Entropy falls as a variant fixes. NTEDS plays the same role in neural space, falling entropy around one parent's trait basis signals fixation, that is, trait dominance. How to read the 3D figure? Access: layer index by depth, kappa, spectral curvature along the floor, and thermodynamic length vertically. Bars: blue bars mark Europe dominant moments, green bars mark North America dominant moments. Bar height is the magnitude of dominance, entropy weighted. Dashed magenta curve: the offspring's nDNA path threading through the dominance field. What stands out in this example? Europe versus North America with offspring. Early layers, short, mixed bars, token and shape traits are shared. Middle stack, about layers 23 to 27 blue bars are taller and more frequent. Europe's traits temporarily fixate while curvature and length are changing. The offspring path runs through this blue corridor, showing biased inheritance during recombination. Upper layers, bars shrink and alternate. Dominance relaxes toward balance as the child settles into its own controller. Why it matters? Asymmetric dominance is where capability and value slant get baked in. If you want a fair hybrid, regularize or add counterpressure exactly at the high NTEDS bands. If you want targeted steering, use those bands to imprint. For distillation, persistent dominance predicts semantic narrowing in the student. Takeaway: NTEDS tells you who speaks when, NTDS tells you who wins overall. Use them to locate, correct, or deliberately shape cultural dominance during fusion. For full definitions, computation details, and additional case studies, please see the book and the website. Enkaryotyping, semantic chromosomal structure, a diagnostic that maps where semantic building blocks live across depth and how they get reorganized by merging, pruning, or heavy fine-tuning. Think of it as a karyotype for models. Instead of chromosomes and bands, we chart concept blocks stacked by layer. Purpose of enkaryotyping: to spot structural edits, duplications, deletions, and relocations of semantic blocks, so we can preserve useful regions, repair missing ones, or rebalance inheritance before deployment. Biological inspiration. Classic karyotyping inspects chromosomes for copy number change or translocation. Our enkaryotype plays the same role for neural semantics, but heights act like copy number of a concept block, shifts across layers look like translocations, missing bars look like deletions. How to read the 3D chart. Layer axis, the diagonal layer index shows depth through the stack. Model axis, 3 groups, parent A, Australia, parent B, Africa, and their offspring. Vertical axis, semantic trait presence taller bars mean a stronger concentration of that semantic block at that layer. Colored curves above the bars, the latent trajectory each model follows through the same region. Compare bends and altitude to see how presence and geometry covary. What stands out in this example? Australia, Africa, and their offspring. Early mid layers. The offspring redistributes presence rather than simply averaging. Some Australia-heavy blocks shrink while Africa-heavy blocks are retained or moved. Mid layers. Consolidated plateaus appear in the offspring where one parent had scattered bars. That is a structural duplication of a blended concept block, useful for stable mid-level reasoning. Later layers. Selective thinning shows where decision-level traits were pruned or reassigned, signaling a cleaner, more specialized headspace. Why it matters. Structural reorganization determines how the child will generalize. Offspring bars that exceed both parents warn of overdominance, copy number gain, missing bars mark fragile deletions, large layer shifts are translocations that can break downstream coordination. Use this readout to place adapters, tune merge weights by depth, or protect vulnerable blocks during pruning and quantization. Takeaway. Enkaryotyping is your chromosome map for concepts. It tells you which semantic blocks were kept, which were lost, and which moved, so you can steer inheritance, not just measure accuracy. For more, please refer to the book and the website for full definitions, settings, and additional case studies. Directional inheritance vector, NDIV, a per-layer vector field that shows which parent's inductive bias is pulling the offspring and how strongly. Concretely, at each depth we place a point at the parental midpoint in nDNA space, kappa, L, then draw a vector from that midpoint toward the offspring. The vector's direction encodes whose priors are winning. Its length encodes the strength of that pull. Purpose of NDIV, reveal asymmetric inheritance across depth, where one parent dominates, where the child balances both, and where drift creates its own path. This lets you set depth-aware mixing weights, place adapters only where needed, and diagnose conflict zones before they show up as brittle behavior. Biological inspiration, analogous to gene flow and introgression in population genetics. Just as vectors in a client show which population contributes more alleles at a location, NDIV shows which parent contributes more belief mass at a given layer. How to read the 3D panel. Access, layer index on the rising axis, kappa, spectral curvature, and L, thermodynamic length, on the floor. Trajectories, blue is Europe, green is Latin America, orange is the offspring, gray is the midpoint track. Vectors, red start at the midpoint and point toward the offspring. Longer arrows mean stronger parental pull. Small labels give magnitude. Consistency, when adjacent arrows point in similar directions, inheritance is coherent. When they spin or fan out, inheritance is contested or drifting. What stands out in this example? Europe plus Latin America. Early layers, about 20 to 22 vectors are short and mixed. The child blends shallow encoders with little bias. Middle band, about 23 to 27 vectors lengthen and align, leaning toward one parent's track, visually closer to the green path here. This is the dominance zone where semantic features are actively adopted. Later layers, about 27 to 30 vectors shorten again and align with the child's own path. Inheritance gives way to stabilization of the offspring's decision geometry. Why it matters. NDIV pinpoints where to intervene, reduce the dominant parent's weight in the midband, add a small bridge adapter on that side, or freeze the stable tails. It also flags layers where inheritance is incoherent, a common precursor to off-distribution wobble. Takeaway, NDIV is your flow compass. It tells you who is steering the child at each depth, how strongly, and when the child stops inheriting and starts thinking in its own stable coordinates. For more, please refer to the book and the website for full definitions, settings, and additional case studies. Epistemic Plasticity Index, NEPI, a per-layer readout of how much a model can still learn without breaking. Think of it as the layer's headroom for alignment. Low NEPI means the layer is stiff, high NEPI means the layer can adapt with small updates. Purpose of NEPI: locate safe and effective adaptation zones for instruction tuning, safety alignment, or task retargeting. Train where NEPI is high, tread lightly or freeze where NEPI is low to avoid geometry collapse. Biological inspiration, akin to developmental plasticity in biology. Some circuits remain malleable under new signals while others are canalized. NEPI measures that malleability in latent space. How to read the 3D panel. Access: layer index along depth, spectral curvature on the floor, thermodynamic length on the vertical axis. Trajectories: blue for Europe, parent A, green for North America, parent B, orange for the hybrid offspring. Upright bars: the NEPI magnitude at that layer. Taller bar means greater capacity to update. Red numeric labels mark especially high spikes. What stands out in this example? Europe plus North America with hybrid offspring. Lower mid-stack, about layers 21 to 24, clear plasticity spikes for both parents, with the offspring showing moderate but usable headroom. This is a good zone for light instruction tuning or safety adapters. Middle band, around 25 to 27 plasticity dips while curvature shifts, signaling a stabilization phase. Updates here should be small and structure-aware. Upper stack, near 28 to 30, the offspring's bars are shorter than at the bottom, indicating consolidation. Large steps here risk brittle behavior. Why it matters. NEPI turns "where should I fine-tune" into an evidence-based decision. Use it to place LoRa ranks, adapter depth, and learning rate budgets, combined with NGDI or NDIV to avoid pushing on layers that are both dissimilar and non-malleable. Takeaway: NEPI is your alignment headroom meter. Train where the headroom is tall, tiptoe where it is short, and you will adapt faster with fewer regressions. For more, please refer to the book and the website for full definitions, settings, and additional case studies. Cultural conflict loss, NCCL, a layer-wise score of ideological misalignment between two models. It asks, when the prompts touch values, norms, or duties, do the model's internal belief vectors push in the same direction or pull apart? Low NCCL means compatible priors. High NCCL means tension that can surface as contradictory judgments, tone shifts, or policy flip-flops. Purpose of NCCL: to localize the layers where cross-cultural fusion will argue with itself. Those are the spots to rebalance with adapters, soften the blend schedule, or add culture-aware constraints before merging, distilling, or deploying side by side. Biological inspiration, like immune incompatibility or hybrid sterility, to lineages carry antagonistic alleles that cannot co-regulate peacefully. NCCL flags these antagonisms in a model's regulatory geometry. How to read the 3D panel. Access: depth is the layer index, the floor axis shows spectral curvature, how sharply the manifold bends, and the vertical axis shows thermodynamic length, epistemic work. Paths: blue is North America, orange is Middle East, and the dashed gray curve is a midpoint reference. Vertical bars: the NCCL spike at that layer. Taller means stronger conflict. What stands out here? North America versus Middle East. Conflict builds from roughly layers 23 to 27, with a sharp spike near 25 where the Middle East trajectory climbs and bends while the North America path stays flatter. After 27, conflict drops but does not vanish, with smaller spikes near the top of the stack. The dashed midpoint drifts away from both parents, hinting that a naive compromise will not be stable. Why it matters. Layers with high NCCL are where merges fracture, distilled students inherit inconsistent policies, and on-the-fly ensembling disagrees of distribution. Mitigate by depth-aware mixing, culture-conditioned adapters, balanced prompt curricula, or per-layer normalization retuning. Takeaway: NCCL is your ideological seams detector. Flat bars signal coexistence, spikes warn that beliefs clash and require targeted reconciliation. For more, please refer to the book and the website for full definitions, settings, and additional case studies. Neural cultural inheritance, final takeaways. Cultural fusion is not neutral. Some lineages imprint, others absorb. Our triad of readouts, NHD and NGDI for drift, NTEDS for pairwise dominance, and NTDS for global attractor strength, lets you see where a merge will move, which parent will steer it, and how hard it will pull. What the table tells you at a glance. Divergence: panel a high NHD and high NGDI flag pairings where the offspring's geometry departs strongly from a parent. The closer parent column names the lineage that anchors the child. Dominance: panel B, NTEDS average dominance rankings show who usually imprints into parent fusions. In our runs, Africa, Middle East, Asia, China, Latin America, and Europe imprint more often, Australia and North America more often yield. Global Pull, Panel C, NTDS total dominance scores identify cultural attractors. High NTDS cultures set the tone across many merges. Low NTDS cultures need protection if you want balanced children. Design guidance. Before you merge, inspect NHD and NGDI by layer. Where they spike, plan adapters, gentler mixing, or brief local fine-tuning. During the merge, if one parent has high NTEDS or high NTDS, allocate more blend weight or geometry-preserving loss to the other parent in mid-layers to retain minority traits. After the merge, recheck NHD and NGDI. If late-layer drift stays high, expect brittle reasoning off distribution and add light alignment or instruction tuning there. Takeaway. Use NHD and NGDI to spot where drift occurs, NTEDS to learn who dominates in each pairing, and NTDS to forecast how strong that pull will be. With those three dials, you can steer cultural fusion toward offspring that are not only accurate, but reason with the values and nuance you intend. For full definitions, procedures, and extended results, please see the book and the website.